For neurological diseases, the biggest barrier is I can't ask a live person to donate their brain or let me take a peek at it because I want to understand schizophrenia or I want to understand Alzheimer's. I cannot really ask you to cut open your brain and show me some brain sample so I can look at it and understand how the disease is developing. Organoid tissue engineering comes from the idea of generating organ-like structures from induced pluripotent stem cells. So the idea is if I take your hair cell or skin cell or blood cell, I can reprogram it or think of it, I can redirect it to become a stem cell. Now your brain or your hair cell or skin cell carries your genetic signature. Now that stem cell will have your genetic properties. After the stem cells, we can tissue engineer them into various organ-like structures. Any tissue you can name these days, these tissue engineers such as me can make them. And that's a great place to start drug testing and personalized medicine. The organoid and tissue engineering gives us the opportunity to develop from scratch a human brain in a dish. We can follow the developmental trajectory of it. We can see how the disease develops. And once we know how the disease develops, we can find drugs or we can find any kind of therapy. We're the tool guys would say, the engineers have given you the tool to assess the drugs or whatever medical device or whatever you made on an actual human tissue. So we give you the preclinical, clinical trials in the sense of the way. For example, if I am looking at a schizophrenia patient-derived brain, then if a pharmaceutical company has a potential drug or they want to test it on and they think it's like one of their top 10 candidates for schizophrenia, they can test it. Another example could be someone wants to test gene therapy or they have this particular gene therapy for, let's say, Alzheimer's or Parkinson's and they want to test it, or an antisense oligonucleotide. They have those made, but they don't know if it actually will work. They've tested in mouse, they have the best candidate, but they don't know if it'll actually work in the human. What concentration would it work for humans? And then we can test it with our preclinical models of these brain organoids and give you an answer. What I see as future, let's say in 5 years or 10 years, we're working on applications of biocomputing. So why not use an organoid for biocomputing, for actually replacing the computer hardware with a biological hardware. The human brain is the smartest that we know of, so we've made the computers. Why not? It can become a computer. So that's one. There's the direction that we're going in. Personalized medicine. So that's another one. We can actually find the drugs for you. You don't have to wait in the doctor's office and think of, oh, will this drug work for me and let's test it out for 3 months and be a guinea pig. We can actually test it and give you the doctors the short list as well.